So first of all, monocots don't have secondary growth, or at least mostly don't. Where's my pencil? So, and that's because there's no ring of like expansion around the outside of a monocot stem. So monocot stem, you got your stem, and then you've got sort of these distributed bundles of phloem and xylem, phloem, xylem, phloem, xylem. And if you were to zoom in on those, these would be phloem cells, and then there would be larger xylem cells here towards the inside. So there's a bunch of these distributed bundles around a monocot. So no cambium. The cambium is a ring of tissue. So on a dicot and in gymnosperms, they have tissue in the center that is essentially, well, it's called heartwood. Even the smaller green stem dicots have this, although it's significantly less of this. But there is like a there's still sort of a heartwood in the center. Okay, then wrapped around that are the most recent xylem. The heartwood was xylem as well, so it gets compressed. The old xylem gets compressed and pushed into the heartwood. So you've got xylem cells. Oops, what just happened? Around here. These are like larger groupings of xylem cells, and if you were to like zoom in on them, you'd be able to see that it's like bundles of xylem. Okay, and then outside of that ring is another ring of cells. This is called the vascular cambium. So the vascular cambium is like what's the equivalent in humans? Kind of like stem cells. So they're rapidly growing and they're producing new cells. So that's like sandwiched in the middle, and then on the outside of that you have the phloem cells. I should have color coded these to make this more clear. Okay, why don't I color these blue for water? Okay. And then we've got our rapidly growing cells here. In the middle, this is the vascular cambium. And then outside of that, we've got the sugar transporting ring of phloem. Okay, so the vascular cambium sort of runs in a band. And these basically continue around the outside of the plant. The configuration of this is slightly different depending on the plant. I mean, they're not identical, but they roughly follow this pattern. And then just outside of that is another band that goes around. That's the other cambium, is the cork, not the cork cambium, the bark cambium. Bark cambium, right? What's it called? Cork cambium. It is called cork cambium. I'm mixing them up because the cork cambium produces the bark. So we've got another cambium layer here of rapidly growing cells, and on the outside of that you've got your bark. Right here. The on green stemmed dicots, you don't have that because there is no bark on green stemmed dicots. So that's these outer two ones here are only found in woody stemmed dicots and gymnosperms. This same layout is more or less the same for gymnosperms. There's slightly different arrangement of the way that they're like put into bundles sort of around the outside, but there's still the same ring structure. And so because they operate and they grow in these rings, the rings can grow and move outwards. We've got new xylem. The old xylem is getting compressed. The old xylem here is going to get compressed and pushed down into the heartwood. We're going to form new xylem. We're going to form new phloem as we move outwards. Now, great question. The old phloem cells eventually make their way this way. Now, this is actually, I'm not 100% sure how this process works. The old phloem cells, which are in orange here, eventually migrate their way inward as the tree grows outward, and they become xylem. They basically become cored out phloem cells, which become new xylem. So I'm not sure exactly how that works. That's a fascinating question. That I'm going to have to investigate. That is the layout, though. That's the layout of this. I should label these so this doesn't just look like a bunch of nothing. This is vascular cambium here. And then you've got xylem on the inside. So the xylem cells are always on the inside. You find the same thing, by the way, in monocots. So monocots will have their xylem cells towards the inside, and they will have their phloem cells towards the outside. The phloem cells are a little bit smaller, like that. So they're always oriented that way. So if this same thing was found on the other side, like over here, it would be oriented like, oops, be oriented like this. Normally, we would take a look at these under the microscope so that you could take a real close look at these and actually see how these structures are laid out. But they generally follow this pattern. This is xylem. This is phloem. And here we have the cork cambium. And this is only for woody stems. Ella, did that answer your question in terms of like how these are laid out around the ring? I'm sure there's a picture of this in the notes somewhere. There's a monocot one. So you can see the individual bundles there. The larger empty spaces there are the xylem cells, and the smaller ones towards the outside are phloem. And there's no vascular cambium in monocots because they don't grow in a ring structure like that. This is roots. Yeah, here we go. See, so, okay, I've got two conflicting diagrams here. This one is saying that the vascular cambium is a ring around the outside, and this makes more sense to me. This makes more sense to me. And this one doesn't. This one says the opposite, that the vascular cambium is in between. Yeah, I'm not sure that's accurate. I don't think it actually is in between. I'm going to have to investigate that, actually. My understanding was that it was actually laid out like this, but then I was looking at this diagram yesterday, and I was like, oh, maybe it's in the middle. In the middle doesn't make sense, but let me explain why one makes sense to me and the other one doesn't. Okay, so my understanding of this process is that you've got. So I'm going to actually switch these. My understanding of this process is that you've got a vascular cambium layer that makes new cells. Then you've got the phloem layer, which is just towards the inside of that. And those are the active cells that are being used for pumping. Okay, living tissue. Phloem is living cells that pump materials, sugars mostly, through the plant. Okay, and then the xylem cells are old phloem cells that have sort of been cored out, and now they're being used to transport water. So there's a transition that happens. I don't exactly know how that transition occurs. It's kind of beyond the purview of this course. You don't really need to know that for now anyway. But that would be something you'd figure out in maybe a botany course. But then so the old phloem cells are getting pushed towards the center and becoming xylem, and then you've got sort of like a layer of active xylem. That is where all the water is flowing within the plant, And then as those get compressed towards the center of the plant, they become the new heartwood. And eventually they just become structural, so they're not really a living part of the plant anymore at that point. So that actually makes more sense, that the cambium is in the center, and that maybe the vascular cambium and the cork cambium overlap with each other. That makes more sense, that there's basically one cambium layer growing in both directions, one towards the cork building cork, and one towards the phloem building phloem. That's what the one diagram says at the top, the micrograph, which makes the most sense to me. Now, it is also possible, by the way, that both configurations exist in nature. And again, this course is this is a very broad intro to botany, and so all the nuance of botany, it's not something that's part of my tool set. I've got a good foundation of botany understanding, but if we start digging really deep into the details here, this is by far and away this is my weak spot in biology. So micro evolution, ecology, pick a subset of biology. I've done it all, but except for botany. So botany is sort of just like, that's a hobby. So I'm not 100% sure whether this configuration is variable. But anyway, the basics are there. Did I answer your question? Okay, all right. Sorry, you already said yes. I'll move on. I have a feeling that this configuration of layers may differ somewhat between dicots, but the basics are always the same. So, okay, what are we up to today? I'm gonna move on. Maybe I'll do some more digging if I can find a better answer to that. But we are on day 17. We're going to be looking at the transportation mechanisms in plants in more detail. So we're going to separate things out a little bit to start. We're going to talk about how things are moved in the phloem. Well, actually, we're going to start with xylem. So xylem is, in my opinion, the most straightforward because it's basically just straws, empty tubes in the center of the plant. And the basic mechanisms are the same for monocots and dicots and gymnosperms. How a xylem works is basically the same. And the purpose of a xylem is to move water from the root to the leaf, because that's the main place where water is being used in the plant for photosynthesis purposes. It is one of the ingredients in photosynthesis. So this video, this Veritasium video, it's a fantastic video, talks about the physics of how to actually get water from the bottom of a plant to the top of the plant. And they ask a really, really cool question, which is basically, if you were pumping, because some of this, by the way, is force that is being pulled from the top. When water evaporates off the surface of the leaf, it actually creates a little bit of a vacuum behind it, and that vacuum pulls up water through the xylem. That process is called evapotranspiration or transpiration. You might have talked about it when you talked about the water cycle way back in grade 8. But that it actually creates negative pressure at the leaf, pulling water up through the xylem. However, and this is something you may or may not be aware of. I don't know if you actually even talk about this in physics. Do you guys talk about the principles of like pneumatics and hydraulics in grade 11 physics? I can't remember if that's grade 11 or grade 12. That might be grade 12. Anyone taken grade 11 physics? Pneumatics and hydraulics? Okay. All right, fair enough. So it's not in the grade 11 curriculum. I'm fairly sure it's in grade 12 then. It's in one of the two. But anyway, what I'm trying to get at here is that when you create a vacuum and you start pulling liquid behind the vacuum, basically you're sucking stuff up with a vacuum. You know, like when you suck on the top of a straw and you like create a vacuum or negative pressure anyway, and you start pulling water up through the straw. You've all experienced that before. I'm sure you used a straw before. Well, there is a, basically, if you want the water to go up higher, you have to pull harder, right? I mean, I guess that just makes sense because you're fighting more, more of a higher force. It takes more energy to raise it up because you're giving it more gravitational potential as you pull it up, right? It takes more energy to lift it higher. I think that probably just makes sense. The maximum amount that you can pull on something by sucking like that is creating a vacuum. That means that you've removed all of the air above the liquid. You've created a vacuum. Well, even if you have a vacuum, which would be the absolute maximum amount of force, there is a maximum height that you can pull water up with a vacuum. And trees can actually grow significantly higher than the maximum height that you can pull water up. So there has to be some kind of unique physics going on inside the plant in order to actually get this to work, because there is no way to pull water up to the height of like the tallest trees. It can't be done by simply sucking the water up. So something else is going on here. And they talk a little bit about it in the video. There's also, by the way, a follow-up video you absolutely don't have to watch. But if you're interested in that hydraulic and pneumatic principle, the idea of there's a maximum vacuum, I think they link it in this video. There's a whole video just explaining that concept. So if you're interested in that, they go into that in more detail. I thought it was fascinating. But anyway, so this is talking a little bit about the laws of physics and how trees and plants, it's mostly trees because we're talking about really tall plants in this case, but how they sort of bypass, they do a little finagling in order to actually get the water up to the height that it needs to go in a tall tree, which is really fascinating. And it talks here about the 3 mechanisms of water transport in the xylem. Okay, so it's going to mention the 3 mechanisms in passing. You don't need to write anything down when you're watching this video, but take it all in because it's really cool physics behind it. Now, in the lesson, we're going to go through those mechanisms in detail. Okay, so there are 3 mechanisms. I mean, I can tell you what they are. They're root pressure, evapotranspiration, which is the sucking from the top, and then the other one is called it's due to the water adhering to the inside of a tube. Water just likes to stick to the inside of small tubes because it has surface tension, and there's a name for that. And what is the name for that? I could describe what the principle is, but I can't remember the name of it. Where is it here? Transport. Capillary action. There we go. Capillary action. So root pressure, capillary action, and transpiration or evapotranspiration. Okay, so the video mentions them in brief and talks about one of them in detail, the physics of one of them in detail. In the note here, I'm going to go through those and explain them in a little bit more detail, how they work. And then the second component of this is we're going to talk about how sugar gets moved around in the plant. So the xylem are not just an empty straw. Sorry, no, I better not mix these up. The phloem are not just an empty straw. The xylems are empty straws. The phloem are not just empty straws. They are living cells, and they are used to transport sugar, and they have to pump the sugar. And the reason why that process is so different, what has just happened here? The reason why, it doesn't really matter. The reason that process is so different is that water, you only need to go in one direction. You only need it to go up from the root to the leaf. But sugar is highly dependent on time of year, even time of day, where you want to send a particular sugar. So let's say it's early spring, you're building buds, you're building leaves. You got to pump the sugar from wherever you've stored it to the leaf. Or maybe it is middle of the summer, okay, height of photosynthesis. Then you're going to be always pumping the sugar from the leaf where the sugar is being made to a storage area. Maybe it's in your stem, maybe it's in the root, wherever it happens to be for your type of plant. Okay, it's different depending on the type of plant. So So because you need to be able to sort of switch it up and like send the sugar wherever you want, it has to be an active pumping mechanism. You have to be able to like basically choose where you want to send it and pump it there. So that's quite a bit different than the xylem system, which is always going in the same direction. Anyway, so we're going to go through those processes, explain how they interact with each other, because there's actually a little bit of interaction between the two. Can't figure out why this thing is going cuckoo bananas today. Why is it doing that? There we go. So we're going to talk a little bit about the interaction between the two systems. They do interact with each other a little bit. So that's in the first half. Then there are a couple questions from the textbook and a little EXA quiz to go along with it. May or may not take the entire first block, probably a little bit less than the first block. Okay. When we come back at 11:05, I'll take attendance, and we are going to do a flower dissection. If you have a flower available to you, awesome. That would be helpful because you can look for the same structures in your own flower. Now, ideally, if you have a selection of flowers, which I have absolutely no idea what you have at home, you might have nothing, and that's totally fine if you have nothing. But if you have a choice, what would be very helpful would be if you had a perfect flower. So yeah, of course, a perfect flower. But a perfect flower has a meaning in biology. A perfect flower is one that has the male and the female parts on it. So not every flower is like that. But ideally, you're looking for a flower like this. And the bigger the better. It'll be easier to see the parts. Of course, you can't see the female part on that one at all. Where's a better example? A lot of the ones that are being shown here are not perfect flowers. They're just, these are just things like people are saying, this is the perfect flower, not a perfect flower. So funny. A few of these are perfect flowers, but they are, yeah, here's a good example. There's a perfect example right there. So in the middle, that structure in the middle is the female part. And then around the outside, those structures are the male parts of the flower. It'll be the easiest for us to identify all the flower parts if you have a perfect flower. Now, there are other perfect flowers where the parts are not as obvious. So, for example, this is also a perfect flower. Those little middle pieces in the center there, the little middle guys, are female parts. And then around the outside here, the little stocky parts are the male parts. So this is actually a perfect flower as well, although it's a little bit more difficult to see which is which. I'm going to see if I can find examples of both of these. I don't have great big giant flowers around my house. I did a little walk through the forest, and this is not a great time to find wildflowers. So I didn't actually find a lot in the forest. But anyway, I'll work with what I got. There's some flowers out on the porch. So whatever you happen to have, even if it's not a perfect flower, we'll be able to at least get some sense of what's going on inside. If you have a pair of fairly good scissors, that would probably be helpful as well. If you've got a nice, really big female part in the middle of your flower, like this flower does, you may be able to cut it in half and actually see the ovules, the egg cells inside. But maybe not. I don't even know if I'll be able to do that on my end with what I've got out on the porch. So surprisingly, I don't have any scalpels or anything at home right now to do a really fine cut here. So yeah, I'm kind of stuck. Anyway, we'll do what we can. But anyway, I'm going to do a flower dissection and go through that. Where are we here? If you want, before we get started with that, you can read the introduction of flowers on page 14 in the course notes. Eventually, you're going to read page 598 in your text, and you're going to fill in the information on the flower dissection sheet. Basically, it's just describing what is the function of the various parts of the flower. We're going to talk about it during the dissection as well, but this gives you a little bit more detail, and then you're going to identify the different the functions of the different parts of the flower. And then for the second half of block 2, I have a really cool documentary that talks about the evolution of flowers, why they're culturally important to people, the co-evolution of flowers and pollinators, some of the really cool things that set angiosperms, which are flowering plants, apart from gymnosperms and why flowering plants are the most common types of plants on Earth. This is one of those documentaries that I would just watch for entertainment. It's such a good documentary. It's a 3-part series. The first part and the third part are also excellent, but they're not related to flowers. So I have the second part here. There's no questions to go along with it. However, I do have 2 questions that I'd like you to consider while you are watching, and they are listed. Where's flowers? Is it here? Yeah, we're not going to... No, yeah, that's not it. Where is it? I've modified this plant reproduction note, by the way, to make it a lot more straightforward than this. Where is it? Hold on. I'm actually just going to open up the new course notes here because it's definitely in the version that I've posted for your. Here we go. This is the version you guys have. Roots, transport, flowers. So there's the intro to flowers that you're going to read. It's on page 14. Here's the part that you're going to fill in. Here we go. Here we go. So there are 2 questions that I want you to consider here while you're watching the documentary. 1, how does the reproductive strategy of gymnosperms, conifers, differ from angiosperms or flowering plants? They're going to make a number of comparisons between those 2 in the video, and you could just answer right on here. Okay, how are they different from each other? And the second component is, what is the advantage that flowering plants offer in terms of reproduction? Why are they so much better? Why are 95, well, that might be a little bit extreme, 90-ish% of plants flowering plants? I mean, there's got to be an evolutionary advantage to them. And so they actually mention 5 or 6 things throughout the documentary about the advantages of flowers, and so I just like you to try and note them down there. Think about those questions as you're watching the video. There are a couple questions from the text that go along with it, and there's a little exit quiz at the end. I will be available during the Google Meet. I had that crossed out because I had to. I was away. Last time I did this and I wasn't available for the Google Meet, but I will be in the Google Meet today. So if you have any questions, you can always drop by. Okay, guys, again, back at 11:05, and I'll let you get started on the first part here. Start off with that Veritasium video. Good luck. Welcome back, guys. If you are here for the second half, if you wouldn't mind, please let me know in the chat. All right, cool. I got everybody. Okay, so I'm going to get my microscope set up here. I have 2 example flowers that I pulled out of some flowers from a little planter on our porch. Unfortunately, these are the only ones that I have available, although they're okay. They're fairly good examples. And in fact, I'm pretty sure both of these are perfect flowers. One of them I can see is a perfect flower. And then the other one, we're going to have to take a look inside just to double check. But I've got this one right here. This one is the one that I can see is a perfect flower from looking inside. And then I've also got this one right here. I think is this a begonia? Is that what this is? Yeah, begonia? Petunia. Petunia. Clearly I'm not the gardener of the family here, but this is a petunia. Petunia. This is a petunia. And this one actually has really interesting markings on the flower. All of the flowers have this, where they're it looks like they're like half one flower, half another flower. I'm not 100% sure what's going on with that. This may be some form of incomplete dominant, or actually this would be codominance, because we have expression of 2 different alleles here in the flower. It looks like there's like a pure white allele for the flower and like a multicolored allele for the flower, and they're kind of mushed together, where the flower is like half one, half the other, and all the flowers are like this on the plant. So I wonder if this is of some form of codominance that's being expressed, which is kind of cool. I'm pretty sure that's not how common petunias are organized. I'm gonna have to take a closer look on the inside of this flower, but it is also possible. Oh, that's interesting. Oh, that's very interesting. In both of these, in both the examples I have of this, the multicolored one, actually maybe this is normal for petunias. The multicolored side has the female part inside of it, contains the carpel, and then the other side, the white part, is the part that contains the stamens, the male part of the flower. I'll try and show you this a little bit closer with the microscope, but I wonder if that's common for all petunias. I'm not actually sure. I'll have to look that up. The other one is a normal perfect flower, where the female part is actually in the center. We'll look at it closer in a second, but the female part, the carpel in the center, is centered, and then the stamens are around it. I don't have any examples this time of perfect flowers where there are a whole bunch of carpels in the center, the female component. But those types of flowers are very common too. I'll show you some pictures of them, but I don't have any to show you under the microscope this time. So let's take a look at these. So you depending on what you've got, if you have your own, you may have one where there are multiple carpels, or you may have one where there's just a single carpel. So both of these just have a single carpel, and I'll show you what that looks like when there's a single carpel. But if you've got something like a sunflower or a dandelion or a daisy, all of those have multiple carpels in the center. So anyway, let me get the microscope out. I'm going to get set up, and then I'm just going to show you the different plant components. Some of them I'll show you on this camera up here. It's not great at focusing really close up, and the microscope is not great at focusing in the sort of medium distance. So we'll do our best. Give me 1 second. I'm just going to grab the microscope. It's a pretty cool microscope. Plugged in and booted up here. Got a little thing set up for the microscope. Hopefully that doesn't, the only thing that affected was change the main screen. Yeah, it looks like it did. So, okay, why is this not booting up? The microscope is on. Why is this not working? Oh, there she goes. Okay, so down, what you're seeing right here is the view with the microscope. Right now, there's a lens on there with 100 times magnification. I might have to drop it down a touch. We can try, I have a 400 times magnification lens as well, but it's a little finicky because it's not on a traditional microscope stand. And if you guys remember using the microscopes in grade 10, if you had an opportunity to use them, when you're on 400 times magnification, even small little movements are like hugely magnified, and so it doesn't work super well. So anyway, we'll give it a shot. I'm gonna actually start on 100 times magnification. I'll try and show you these parts. Just I wonder if I can use the camera on my iPad to give you sort of like an intermediate view. Give me 1 second. I might be able to do that. Okay. So what you're seeing on screen right here, well, nothing at the moment, but I'm going to throw this flower underneath so you can sort of see the different parts. So first of all, I'm going to start at the base of the flower. So there are some sepals down here at the bottom that are sort of modified leaves. You can see, there you go. You can see them underneath. Those would have originally made up the outside of the bud that contained the flower. And they're just protective. So they're there to protect the flower before it blooms. there is a receptacle that is sort of at the base. Let me see if I can find where the receptacle is on here, so you can see a little bit better. Right there, sort of where the sepals are coming together at the bottom is the receptacle. Pull this down. There we go. You can just see right at the bottom there is the receptacle. There's like a little membrane at the bottom. And that eventually that's going to dry out. Once the ovary, the ovules inside the ovary are fertilized, they're going to become seeds, and then the ovary is going to form into a fruit. So all flowering plants form a fruit of some kind. It's not all edible fruit, like an apple or something like that. Some of them just have what looks like a seed pod. That seed pod is fruit. That's like the technical term for that. So for example, if I showed you, I'm going to jump back and forth here, but if I showed you like a fruit from a rose plant, I believe they're called rose hips. It's the fruit from a rose plant. So they look like this. You wouldn't want to eat that. That's not an edible fruit, but it just basically is forming like a sac, a protective sac around the seeds. Sometimes that sac, as you'll find out about in the documentary, sometimes that sac is to lure in animals to eat it, and so it contains a lot of extra sugar. The idea being there that the animals are going to actually spread the seeds to a new location, which is very advantageous for the plant. Basically, its offspring get to be distributed far away from the parent. In general, plants don't want to grow their offspring right adjacent to them because then they're in competition with their own children. So, should you be taking notes? You don't have to, Andrea. I am going to be discussing the function of sort of these like major tissues of the flower, the components of the flower. But it's all in the reading from the textbook, which is on page 598 as well. So, and you're just gonna like skim through 598 afterwards anyway and add them in to the sheet. So, I mean, it's up to you. You can fill them up in as we go because I am going to discuss the function of each of these. But I'll leave it up to you. You don't have to be taking notes while we're doing this. So, anyway, the receptacle at the bottom there, this is what I was trying to get at here. The receptacle, which you can just see, it's kind of hard for me to point to it on here, but it's like just this area right down here at the bottom. I should get a little pointer. I'm going to use this. Boop. This is the receptacle right here. That part is going to dry out once the fruit is formed, and then it'll allow the fruit to detach from the plant. So that's sort of what the receptacle does. Right now, it's firmly holding the flower, and then later on in the life cycle of the plant, it's going to drop the fruit. So you've probably experienced this if you've ever gone apple picking or something. If you try and pick an apple, if the apple's not yet ripe, the receptacle is not dried out yet, and so it's effort. You have to like basically twist the fruit off. But if the fruit is fully ripe, the receptacle is basically releasing the fruit at that point. And that plays an evolutionary purpose as well, because the seeds are not actually ready to be taken until the fruit is ripe. In other words, they're not fully developed and they're not ready to be taken away and distributed. And so the plant basically lets the animals know that could potentially consume the seeds when it is time for them to be taken. And it does that in multiple ways. So one of them is that it releases the fruit through drying out the receptacle and releasing the fruit. And then one of the other ways it does it is with color change. And so there are certain colors that are really attractive to mammals and other fruit-eating animals, although it's largely mammals and birds. And they are highly attracted to certain colors. Red would be a good example. And so fruits have a tendency to turn those colors, signaling to animals that they are ripe. And that's what ripeness is. They also change the flavor and texture of their fruit to entice animals to come and take the seeds, which is why an unripe apple tastes terrible. I mean, it doesn't taste great, and that's because the sugars that are contained within an unripe apple are not simple sugars. They're not the sweet sugars. And that's intentional because the plant does not want to attract something to come and take the fruit away yet. So when it becomes ripe, it's going to convert a lot of those sugars to simple sugars, which will make it taste sweet, enticing an animal to come and take the fruit. So there's like a benefit. Hi, are you guys joining me today? You can join me if you want, Leo. You want to? Come on over. Come on over. You want to say hi to my class? You don't want to. You don't have to. You just, if you're over here, you're out of the frame, okay? You can come and see what's going on. You don't have to get in on the video if you don't want to. Okay, so anyway, that's the bottom of the flower. We talked about sepals, receptacle. Now let's get to the actual sort of like the sexual organs of the flower. So I'm gonna take off the petals. Now the main function of these petals here. Do you remember what they are, Liam? What do the petals do on the flower? You're not sure? You don't remember? That's okay. You don't have to be nervous. So they obviously have patterns on them, and then those patterns are to lure in pollinators, to get pollinators into the flower. They're basically like a bright sign. They have other patterns on them that are not visible to humans. We're not pollinators. And many of those patterns are, you can see them in ultraviolet. So they're actually reflecting ultraviolet off the surface of the leaf, and a number of pollinators are able to see the ultraviolet part of the spectrum, or at least the close part of the ultraviolet part of the spectrum, like bees. There's a number of insects that are pollinators that are able to see into that part of the spectrum. And so they can see the signs that are evident on petals that can attract them in. That is another thing that's shown in that documentary that you're going to watch afterwards. It's fantastic. They show you a number of photograph comparisons between an ultraviolet view of a flower and a regular light view of the flower, and you can see what the patterns look like in ultraviolet light, which I think is amazing. So anyway, that's the purpose of these petals. I'm going to take the petals off. Now, one of the alternate uses for petals is that it can make the flower only accessible to a particular pollinator. So by being specific about which pollinators you allow in, you can basically corner the market on a specific pollinator. A great example of that would be any kind of flower that has a really long flower that leads in to where the pollen or potentially nectar is stored. The nectar is usually stored in a type of flower structure at the base of the flower. And if you've got a long structure like that, you're only going to be able to attract in insects with a proboscis, which would be like a long little nose that they can use to like pull up the nectar, or something like a hummingbird. So other types of insects are not going to be able to get in there. And therefore, if an insect can only access maybe 3 or 4 types of flowers in a given environment, there's a higher chance that they're only going to be going from your species to your species to your species, and therefore your pollen is going to get transferred to other flowers that are of your own species, which is what you require for pollination. So if you can narrow down which types of pollinators can actually access your flower, that is beneficial to you as a plant, because you have a higher chance of passing your pollen on to a neighboring flower and receiving pollen from a neighboring flower. Remember that the idea of sexual reproduction is that you are able to combine with another member of your species, and plants can't go to the other member, so they have to come up with some creative ways to ensure that the other members are going to get to them. Now, gymnosperms have a different strategy for this. Gymnosperms, remember, are conifers, plants with needles, so they don't have flowers, and their strategy is just throw their sperm up into the wind. Basically, have a sperm explosion, throw their pollen up everywhere. There's billions and billions of pollen. They produce far, far more pollen than flowering plants do. And then they just hope. They just hope that their pollen is going to land via wind on an appropriate mating partner. So it's not that great of a strategy. They talk about that more in the video as well. So, okay, now I've removed the outer part here, and I'm going to show you what this looks like. First, I'm going to show you under just this little tiny magnifying glass, but you can't really see the components too well in here, so I'm going to have to put them under the microscope so you can see them a little bit clearer, because this is a little tiny flower. So right here, you will see in the center, that is the carpel. So that the green thing that you're seeing in the center is the carpel. That's the female part. And the stamen, or the stamens, the male part, are actually grouped around the outside, and they have little pollen sacs on the top. Now, the pollen sacs are different shapes and different colors, as are the colors and shapes of the carpel, totally dependent on species. So I'm going to show you what this carpel looks like up close, so you can see what it looks like under the microscope, and then I'm going to show you the little pollen sacs of the stamens here, so you can see them up close. We might actually be able to see actual little pollen grains on there as well. So, so let's zoom in here. So, in brief, I'm going to pull the sepals off to make this a little bit easier to see. I'm also going to show you another example flower that looks a little bit different from this flower. We can do a little bit of a comparison between the two of them. So let's try and zoom in here. I'm going to switch to the microscope. All right, here we go. Oop. Come on. Right in the center there. This microscope is very finicky. Oh, beautiful, beautiful, beautiful. There we go. There we go. Oh, I got a little bug too. I don't know what that bug was. There we go. Okay, so that little hairy thing on the end there, the green bulb, is the tip of the carpel. What we're looking at right there is the stigma. So the stigma is the top, the sticky part of the carpel. I'm going to pull the male parts off here in a second around after we look at them so that we can see the style, which would be right below here, right below the stigma. And then the bottom, hopefully we'll be able to see the ovary as well. Now this is far too small for me to cut in half. If you've got a really big carpel on yours, I would cut it in half. You may be able to see the ovules inside with the naked eye. On some plants, they're large enough that you can actually see them just by looking at them. They're little tiny spheres that are inside the carpel, or that are inside the base of the carpel, which is the ovary. But here, I mean, I'd be able to see them with the microscope here, but I don't have a tool to cut this in half properly because it's far too small. So I'm going to ask you to imagine that it's in there. It's definitely in there. So anyway, this is the stigma. That's the sticky part. So because this is a perfect flower, you're right, by the way, Andrea. I think it was an aphid that was crawling around there. I don't know where he's gone to, or he or she. It's off on an adventure somewhere. You can see a problem here. Sorry, this is the stigma right here, this part right here. So the problem that you're likely thinking of here is that, hey, if you've got pollen producing components and you've got the female part that's taking the pollen right next to each other, isn't that going to be a problem? Like, isn't this thing just going to fertilize itself? Well, in some situations, yes. So in nature, there are a lot of self-fertilizing plants that just don't care about that. They're fine to self-fertilize. Perfect example, the dandelion does not care. They're happy to self-fertilize, and they don't really have a lot preventing them from self-fertilizing. So they are technically sexually reproducing. They can accept pollen from neighboring flowers, but they often don't. They often just self-fertilize. And so, yes, that is inbreeding. It does defeat the point of sexual reproduction. So my guess would be some previous evolutionary iteration of those plants was not self-fertilizing, but then there was no situation in which it was advantageous to ensure that there was no self-fertilizing, and therefore it just evolved a mechanism that allowed it to self-fertilize, which is totally fine. So some plants do that. There are many plants that just don't care that much about sexual reproduction. We have previously talked about why sexual reproduction is so important. Remember that it creates diversity in a particular organism, which improves its rate of survival, which is why sexual reproduction is so common. And it's also the reason why there are mechanisms in place to prevent self-fertilization. So I'm going to give you one. Many plants, like this plant, don't have their stigma, the top of the carpel, be sticky at the same time that they're producing pollen from their pollen sacs, which we're going to look at in a second. The pollen sacs are on the male part on the stamen. So if they're not active at the same time, the likelihood is that they're going to release their pollen. that pollen will be mostly gone, and then their stigma will potentially be receptive to other plants. And this is something, it's basically random, and so it's likely that a neighboring plant that is releasing pollen is not going to be a close relative, or at least it's more likely that they won't be a close relative, which increases the potential genetic diversity in the offspring here. So, I mean, that's the idea behind it. There are other mechanisms as well, but there are often mechanisms that are preventing self-fertilization from occurring here. So this is the top, this is the sticky part. Pollen is going to land on this part, and the pollen it's really cool. It actually grows a little tube that goes down through the style all the way down into the ovary, and then it will grow into a specific ovule. This is from the pollen. It's called a pollen tube. And once that pollen tube reaches a particular ovule, which is an egg, this is equivalent, by the way, to sperm and egg in human. Once the pollen tube grows into an egg, it's going to send the nucleus down into the ovule, where they will fuse together to form a 2N organism. Keep in mind that the pollen is 1N, it is haploid, and the ovule is 1N, it is haploid. So in order to make a mature seed, a seed that can become a diploid organism, you got to fuse those two nuclei together. And that's because of meiosis, right? Meiosis is creating haploid cells. Plants undergo meiosis just like animals do. So I know I mentioned way back in the diversity unit, I talked about how some plants don't really care about that whole haploid-diploid thing. So you get tetraploids and things like that in mature plants. That is also true. I'm going to discuss the rules, but just be aware that there are many exceptions to the rules. So in general, the rules are that plants are going to do meiosis. They're going to produce a haploid pollen and a haploid ovule, egg. And those two haploid nuclei are going to come together to form a diploid plant. Okay, a diploid seed, a fertilized seed. Now, there is also a second component to this, which is that often there is another haploid nucleus in the pollen, and that one will join up with, or sometimes it's diploid actually, it depends on the type of plant, and it will join up with another nuclei, another potential haploid nuclei that's also in the ovule. A second fertilization occurs. That's called double fertilization, and the second fertilization creates an endosperm. So it's not going to become a plant. It's actually going to become an energy source for the plant. So it grows into the nutritious part of the seed. The endosperm is the nutrition part of the seed. So if you've ever had like a corn kernel, for example, the sweet part of the corn kernel that's like on the outside of the corn kernel, the part that you eat, that you want to eat, is the endosperm of the corn. It's actually an energy source that is like a sort of a separate organism from the seedling. It doesn't become a mature organism. It doesn't become a plant. So its only role is just to grow and be an energy source for the actual plant. And obviously humans don't do this. This is unique to plants. But this is going to come up when we talk about sexual reproduction in more detail tomorrow. I just wanted to mention it. So that is a second component that is happening here. So anyway, this is the stigma. It will be sticky at some point and receive a pollen. The pollen is going to grow a pollen tube down the style to an ovule where it's going to make a fertilized seed. Okay. So let's look at the male part now. So the male part is right next door here. What? Okay, if I go this way, yes, right there. Now let's see if I can get that in focus. Am I getting more in focus or less in focus? Less. I was hoping we'd actually be able to see some pollen on here, but I'm not sure that we will. Anyway, there are a number of these male structures. So the darker part there is actually the pollen sacs for this flower. And a little bit different from the one that's in the textbook, which has it has a filament holding onto it that's like a very thin filament. A lot of flowers have that thin filament holding up the pollen sac on top, or holding up the anther, I should say. The anther contains pollen sacs. So in this case, the anther, this is the anther, it's covered in little pollen sacs. And the filament holding it up is actually fairly robust here. It's actually this big white structure underneath. That's the filament. So some plants have a very thin little filament on top. This one's got a big robust one. Oopsie-daisy. What have I done? There. Yeah. So you can see it. The filament is actually the green part in the center there, but it has this like sort of white, hairy filament that's like, or at least membrane sort of that's on top of the filament here. That's this part up here. So this right here is the anther. So it contains pollen sacs. So what I'm going to try and do here is go up a level of magnification, and I'm going to see if we can actually see some pollen on here. I'm not sure if we'll be able to actually see it, but we're certainly going to try. Okay, well, we'll take a shot at it. This lens this lens, because it's such a high power, it's kind of unwieldy, so it can be quite difficult to get it to focus. I'll do my best. Okay, here we go. All right, now I gotta actually get it focused on it. Oh, we're on the right track. The focus is also really difficult. They put the focus knob right at the tip of the camera, and obviously if you touch that, it moves the camera around a huge amount, and so actually focusing on something is borderline impossible. It's a really terrible focusing mechanism. Oh, okay, we're right on top of one right here. Come on. Oh, shoot. Oh, we're coming into focus. Oh, here we go. Okay. You know what? I actually think that the pollen sacs that are on this anther are not open yet. So this plant might have a receptive it may have a receptive stigma, but it is not, I don't believe it's currently releasing pollen. This looks closed to me. Yes. Usually the ones that I've seen that have active pollen on them they look fuzzy because they have like little bumps all over them where the pollen has come off. So I don't think that these are open. So that's kind of cool. So the stigma might be active here, but these pollen sacs do not appear to be active, and I don't see any pollen on them. So I might have caught this just a touch early. The flowers are blooming, obviously, so they could be open, but I don't believe that they are in this case. Okay, that's kind of cool. I'm going to peel these off the outside of this flower, and hopefully we can see that the lower part of the female parts, which is the style and the ovary. We're not going to be able to see the ovule because the ovule is tucked away inside, but yeah, I don't think that this thing is currently releasing pollen. So that's kind of cool. That's actually the first time I've taken a flower apart and had seen that it doesn't have pollen on the anther. So here we go. So this is a pretty clear view now of the ovary. Sorry, not on screen. I can see it in my hand here. So I have to switch back the lens because you're not gonna be able to see much if I leave it on this high magnification. But on a slightly lower magnification, you'll be able to see it. Okay, so here we go. Take you down to the nothing. I'm looking at nothing. What's this? Well, that's it. Hold on, let me just, if I look towards the top here. Okay, so that up there, let's see if we can get in focus. Gosh, is this ever hard to focus? It bumps and jips and jives all over the place. Oh, it has pollen on it. Oh, that's cool. I'm pretty sure it does. I'm pretty sure it does. Hold on. I couldn't see it from the other view, but I'm fairly certain this already has pollen on it. I wonder if we can zoom in on that with the other one to actually catch the pollen. Why is this so hard to get into focus? Come on. Yeah, I'm pretty sure there is pollen stuck to it right now. Right? I don't know if you guys can see it. No, I want to go the other way. There. So that little bobbly bit there on the top, this is the stigma. So if we slide this down this way, there you can see the style. So the style is this little tube here in the middle. And then this little part at the bottom is the ovary. So that's the ovary. This whole structure right here is the ovary. And inside that are going to be these small circular, I wish I had a scalpel so I could probably cut this in half. It's way too small for me to cut in half with any tool that I have here. But if you crack it open, you'll be able to see ovules inside, little eggs, which are going to get fertilized by the potential pollen that lands on it. Now that I've been able to do this, I'm gonna try and look at it with a higher magnification because I'm fairly certain I can actually find some pollen on it for you. So just give me 1 second. How am I doing for time? I don't want to take up all your time. I got to leave time for you to watch the documentary. So let's see if I can zoom right in on this little guy. For some reason, it is so difficult to get this to work on high magnification. I'm really trying. I have an attachment for this camera to go on our normal. Oh, this is promising. I have an attachment for this camera to go on a. Oh, it actually fell off. The stigma fell off. All right. I wonder if I can get it easier now. Just put it right in the center of the view. Right in the center. Oh, I actually got it stuck to the microscope. Oh, I screwed the lens right off. Oops. Oops. Oh no. Get out of there. Oh no. Oh, I got it stuck inside. It's in the lens. Whoops. This isn't exactly the controlled environment of a laboratory here. This is just on my desk, so all right, let's try this again. Oh! You silly thing. It doesn't help that it's sticky. The fact that it's sticky makes this way harder because as soon as it touches the bottom of the lens housing, it sticks to it. Oh, for goodness sakes. I'm really trying here, guys. Sorry. Oh, we're on the right track. You can see how jiggly it is here. I'm barely even touching it, and it's jiggling like crazy. Oh, come on, give me some pollen grain here. There's got to be one on there. I'm sure. I'm absolutely sure that there's a pollen on there somewhere. No? To be perfectly honest, all this manipulating, I might have knocked all the pollen off of it, but shoot. I see none. Okay, well, we took a shot at it anyway. I don't see any pollen on here, so it's possible it either had pollen on it that I knocked off with all this craziness, or it's quite possible it hasn't been pollinated yet. It may have just bloomed. So let me clear all this stuff out of the way here. So I've covered all the different parts. I just want to show you a different example flower so you can see sort of a different example of what these can potentially look like. Does anybody have any questions about this so far? The parts of the flower and what they do? Let me know if you think of anything. I'm going to start dissecting this other flower. So the other flower that I'm going to do is this guy right here. I'm not going to give you the full walkthrough. I'm going to pull these petals off. And I'm going to go straight for the anther, straight for the stamen and the carpel to show you what they look like. I'm going to pull these petals off first. Now this is an example, by the way, of one of those flowers that has sort of a long tube of petals in order to get down to the sexual organs of the flower. Oh, this one's got lots of pollen. We'll be able to see the pollen here. Cool. The pollen's actually coming off onto my fingers. There's so much, so we should be able to see this a lot easier. Come on, I gotta get this in focus. Whoops. Oh. There we go. Okay, so this is the 100 times magnification. You can see it actually. You can actually see the little dust on there. So that little dust that you're seeing is the actual pollen. Let me slide this up a touch. There you go. Oh, beauty view. Sorry, even just me kind of talking is making this vibrate all around. So these little bumps that you see on here are the actual pollen. So the pollen here is being released from the pollen sac on the anther, and these little green parts down here are the filaments that are holding up the anther. Okay, so those are haploid cells on there. This is essentially plant sperm. I'm going to show you what the stigma looks like on this one as well. It's in the center. It's right there. And it has some pollen on it as well. Oh, and there's a little bug in there. Hey, buddy. I have no idea. That might be an aphid too. I'm not sure what that is. Could be an aphid. And that little bug is a pollinator, helping this little plant out. Oops. How do I want to move this so I can get a better look at the bug? And I have a feeling the bug is crawling up into the microscope right now. Or maybe he went deeper into the flower, but there we go. You can see the stigma right there. So the stigma is, it looks like it's active on this plant. It's sticky. And so there might be some self-fertilization going on in this plant. But there it is in the center. It's the, sorry. Man, is this everything ever hard to maneuver around? I'm doing my best. This is the first time I've ever done a flower dissection and there's been a pollinator inside the flower. That's kind of fun. So it's right. This is the stigma right here. I'm attempting. Come on. Why are you being so difficult? Just trying to get it into good focus for you guys. Close enough. Oh, you can really see the pollen though. Check that out. Right there. Pollen grain, pollen grain, pollen grain. And then here we have the stigma in the center. Sorry, it's a little bit blurry. Getting this thing properly focused is a huge pain. So there you go. So there's a little bit more variety. This flower looks a lot more like the one in the textbook, whereas the other one, the anthers are, well, they weren't open, so the pollen sacs on them weren't open, so you couldn't see this sort of like fuzzy pollen on them, but they also have a different characteristic shape. So that's the basics right there. If you follow this down, if you follow the stigma down through the style to the ovary, what you do down here on the flower, again, I can't cut it open anyway, so it just looks like a green knob. But that is what contains the ovules, the eggs, which are going to get fertilized by this pollen. So, really quickly, I just want to show you some different variations of this. The flower that you're looking at may look absolutely nothing like this. So I'm going to show you some... I'll show you some other examples of perfect flowers and some examples of imperfect flowers as well. So, sunflower is a great example of a perfect flower that doesn't look like a perfect flower. So let's see if I can find a nice high-res image of one here. If you have ever eaten a sunflower, like you've actually like harvested sunflower seeds from a sunflower, you may have noticed that there's like a whole bunch of seeds all across sort of like the center of the plant. And they're sort of like sticking out. Each one of those seeds is in its own ovary. So in that case, each ovary only contains one ovule and is only going to produce a single seed. And some plants have that configuration. I wish I had a better high-res image here. Maybe you can search. Can you search for high-res? Is that a thing? High resolution. Oh, that's pretty good. So here you can actually see around the outskirts here are the male parts of the flower. So they're sticking up. Each one of those is its own stamen, with a filament and an anther on it. And then in the center, these like concentric circles structures in the center, each one of those is a separate carpel. So each one has an ovary at the bottom with a single ovule, and a stigma on the top with a style going down to that ovule. And so each one of those ones in the center is going to produce one single seed from its ovary. So in this case, it's not really much of a fruit. It doesn't really produce a true fruit here. I guess I mean, by the technical term, it is a fruit, but the seed coat on the outside is basically the ovary in that case. It's basically each seed is its own fruit in this case. So your flower may look like this. For example, I think daisies look like this. Let me double check. So a daisy is just like this, where they have a ring of the male components around the outside, and then they have the female components in the center. Each one of these female carpels has its own stigma, style, and ovary, and each ovary probably has one seed in this case, so it's going to form its own fruit. So that's another type of configuration that you might find in nature. You may have pulled one of these flowers from your garden or patio, or if you were able to find a flower, even a dandelion. This is very similar to the configuration in dandelions, so you would see something very similar. Okay, I'm gonna stop talking about this at this point because I feel like I probably made my point here. We've gone through the different structures. What you're going to do now is, what you're going to do now is read the little introduction on page 14 of your course notes. It's what's above sort of like the flowered section, okay? You're going to use 598 in your text, or you can use internet resources, or you can just use what we just discussed here in the dissection to fill in the table. The table is just the different functions of the different parts of the flower, okay, which you're probably familiar with now. But if not, again, you can use that 598, the text. It's a review of what the flower parts do. And then you're going to watch this really cool documentary. So I don't want to take up any more of your time. You'll need the rest of the time to watch the documentary. It is fantastic. There are two questions from the text that go in your questions document, and there's a little exit quiz just to test your knowledge. I'm going to change that little thing at the bottom that crosses out the I will not be available. I will be in the Google Meet today, so I got to fix that. But if you guys have any questions, if you need anything, let me know, and I will be here. And I got to clean up these flower bits that are all over my desk. Okay, good luck.